So this is a lecture on how we study biological aging from August 21st, 2020. And this is the first part of a two-part lecture. And so today we're going to focus on defining certain terms, specifically what a biomarker is, as well as what a cross-sectional and longitudinal study are. I'm going to talk about how model organisms are used to study aging in the lab, and several benefits and drawbacks of doing that, as well as several advantages and sort of disadvantages of using the two types of studies listed above. So the cross-sectional and longitudinal studies, and how they're good and how they're bad to measure rate of aging, particularly in individuals. And so a recap of our last lecture, there's basically two important takeaways from that lecture, which is that defining aging is really difficult, and measuring the rate of changing in an individual is also really challenging. Yet there are still biogerontologists out there studying aging, and there's a whole plethora of aging research going on. And we're still going to attempt to understand aging in order to improve health span and lifespan in individuals. And so how do we do that? And so one of the ways that we can study aging is in the lab, using either model organisms or cell lines. And so there are several different types of cell lines that are used in cell biology research. Some are better for studying aging than others. And so there are what are known as primary cell cultures, which are differentiated or cells that have like a specific fate already assigned to them. There are replicating cell cultures, which are not differentiated and so have the potential to become different cell types. And they're also mitotic or still dividing. And then there are also stem cells, which are cells that have been immortalized in different ways in the lab and can divide basically as many times as they want for the rest of eternity. And a primary cell culture being differentiated usually comes from a living organism. So cells are removed from a specific organism. There are some biochemical manipulations made to them, and then they can be grown in a dish. But because they already are differentiated or they already have their fate, they're not mitotic or dividing, and so their lifespan within the dish is pretty short. And so you can really take out a primary cell culture, do one experiment with it, and then it's gone. And so one nice thing about primary cells is that you can take them from different animals of different ages and look at age-related differences between cells that actually came from an organism. So you might take a neuron from a young mouse and a neuron from an old mouse. You can grow them in a dish and then look for differences between them to start making some inferences about aging. In terms of replicating cells, because they're non-differentiated, they don't particularly have a fate determined yet, but they're still dividing. They're really great for identifying factors that are involved in cell death or cell senescence. And that's because cells have sort of a defined number of times that they can divide before they senesce or die. And by studying these mitotic cells that have this sort of limit of division, factors have been identified that are involved with kind of this cell aging as well as cell death. And stem cells or immortalized cell lines are not particularly great for studying aging, only because they don't actually age, right? And so they're not used a lot in aging research. Stem cells are really important in terms of regenerative research, and regeneration is one thing that sort of relates to aging, right? So the ability to regenerate or replenish some old tissues would be a potential therapeutic treatment for aging and some of the issues related to it. But we can't learn a lot about how cells age from studying stem cells. And cells are great, but they're individual units. And we also want to be able to study aging in organisms or in the whole body, as well as in populations. And so we can also use other model organisms to do that in the lab. Specifically, we can use yeast and worms. And so yeast are unicellular, and they don't actually have protection from their environment. They come into direct contact with their environment at all times. And since we talked on earlier lecture about how important environmental factors are to determining the rate of aging, yeast are a great model to study all of these, the interplay between the environment and what happens in a cell specifically throughout the course of aging. And so over here you can see some of the environmental and also biochemical, both inhibitors of aging in yeast as well as some promoters of aging. And so yeast have been great to start figuring out aging at the cellular level and how the environment affects cells. And then we obviously also want to think about how the environment and how aging affects tissues and a whole organism. And one whole organism that's been beneficial in understanding that is the nematode worm, C. elegans. And C. elegans have a very short lifespan, so they grow from embryo to adult in 3 days, and then they live to about an average lifespan of 20 days. But they have some complicated tissue structures, which you can actually start to see on the outside of this worm here, and those lines, which are hypodermis. They have complicated neurons, they have muscle, and so because they are multicellular and they have these tissue structures, we can ask questions about how intracellular communication and how whole bodies age in response to environment and genetics, and start identifying how the cellular events affect aging of the entire body. And so kind of moving up in complexity, studying aging in yeast or in worms is great, but yeast are unicellular and worms are invertebrates. And so in order to start making more conclusions about humans, we want to study aging in mammals as well. And we can do that in the lab by studying mice and rats as well as primates. So over here you can see a picture of some mice. A and B are actually a young and an old mouse. C and D are a young and an old mouse. But the difference between B and D is that D actually has progeria-related mutations or kind of premature aging mutations involved in it. And so we can use mice and rats because mammalian physiology and cellular function is very similar between mice and rats and humans. And so we can look at the age-related changes sort of in the physiology, muscles, motor function, brain function, as well as in cellular function, and make some conclusions from that. And so you can see here, right, one thing that you might notice is that there's sort of that graying of the hair. That's one of those physiological things that we can study using mice in the lab. And yes, we can also study primates. Studying primates in the lab can be difficult at some times. It's the most sort of ethically ambiguous thing we can probably study in the lab because monkeys are cute, and monkeys are also most similar to humans. They have personalities, and so sometimes studying them in the lab causes a lot of ethical concerns, particularly with some of the animal activist groups. But primates are kind of an ideal lab model for studying aging because humans are also primates. So specifically in aging research, rhesus macaques, which you can see here on the right, are used a lot. And primates are also mammals, and so they have some of those similar physiological and cell functions that humans have. And they do have kind of this time-dependent or age-dependent decline in function that mimics humans' decline in function really well. And so primates like the macaques will have hearing and visual defects over time. They'll lose their motor function, their muscles will atrophy or get smaller, they'll have gray hair. And so there are a lot of different kind of age-related phenotypes that we can study using primates. And additionally, primates actually suffer from some age-related diseases or some kind of time-dependent illnesses as well. Older macaques, for instance, can have type 2 diabetes. They have certain types of cancer. they can have a pseudo form of neurodegenerative diseases like Alzheimer's and Parkinson's. And so they're not only good for studying sort of like age-related decline, but also the diseases related to aging. What's important to remember about all of the model organisms is that they do have a shorter lifespan. And so that's one disadvantage of studying them and trying to relate the results back to humans, because humans have a much longer lifespan. And it's possible by studying something that is either unicellular or an invertebrate or has a shorter lifespan, we're not making accurate conclusions about what we would see in a human. But they are cheaper, easier, faster, and obviously more ethically sound to work with than studying a human in the lab would be. But one way is that we can actually use humans for research into aging is by studying a genetic condition that's known as progeria. And progeria is pretty rare in terms of genetic conditions, but what ultimately it leads to is slow growth and rapid aging. And there are different types of progerias, but Werner's syndrome is the specific type that's been kind of held up as a model for aging. And that's because Werner's patients will age normally to about 20 or 30 years old. And so you can see a Werner's patient here at 15, looking like a kind of typical 15-year-old. And then once they reach about 20 or 30, they go into a period of sort of this slowing of growth and rapid aging. And so this is that same Werner's patient at about 48 years old. And you can see that she looks much older than what you might think a 48-year-old would look like. And we can learn a lot about sort of the genetics of aging from studying Werner's patients. So Werner's is a recessive disorder, and just by studying patients, we have identified the gene involved as well as the protein. So there are mutations in the WRN gene, which affects the WRN protein and basically makes it non-functional. And what WRN protein does is repair DNA. And so this data has supported the hypothesis that biogerontologists have had for a long time, that the mutation accumulation that occurs with age, as well as the inability to accurately repair all of those mutations, contributes to aging. And so Werner's patients have sort of provided some data to back up that hypothesis, that DNA repair as well as mutations contribute to aging. And so we can also study animals in nature and sort of make comparisons between long-lived wild animal species and humans. And so this is a field of study of longevity known as comparative biogerontology, where we look at wild animals with long lifespans and try to see what is conferring that long lifespan onto them so that we can think about how that might affect aging. And we've learned a couple things about aging from studies like this. We know that large mammals tend to live longer than smaller mammals. So this is a graph showing longevity over body mass, specifically in mammals, and you can see that longevity of the smaller animals is lower, whereas the larger mammals have a higher longevity. Humans don't necessarily fall into this linear distribution, and there are some reasons for that, and they're listed pretty well out in your Biology of Aging textbook, one of which is likely the contribution of intelligence to human survival. And we've also learned that the ability to adapt to your environment, avoid predation, and forage, kind of find food, is correlated with a longer lifespan, right? So if you can avoid being eaten in the wild, you live longer. If you can make sure you spread out wide enough to get food that you need to survive, you can live longer, as well as this idea of having safety in numbers or a social structure also being associated with a longer lifespan in the wild. And so all of these things together, we've learned some pretty interesting things about aging by studying species in the wild, as well as by studying model organisms, cells, and people sort of in the lab space.